Now that we see the very general outline of how base pairs can connect together, we can look further into the ultimate 3D structure that occurs, particularly in DNA, when two pairs of nucleic acid strands will then hydrogen bond to form this three-dimensional structure. Watson and Crick and Rosalind Franklin modeled DNA using X-ray crystallography. What they found is that DNA forms a double helix type structure. And actually, it forms it in what we characterize as bDNA. Our DNA, or bDNA, has these major and minor grooves. You can see this is based on the fact that one area is longer and the other is shorter in the internal or groove portion, and has characteristic lengths as outlined here. A whole segment of major-minor groove rotation is about 36 angstroms long. The width of a DNA is about 20 angstroms long. You can also see that DNA rotates in a particular direction. It is left-handed. And again, to remember, left-handed and right-handed, point your thumb up and look at the way that your fingers curl to determine a left-handed or right-handed rotation. You'll see that when you hold up your left hand with the thumb pointed up, your fingers curl in the same direction that the DNA actually curls as well. Now, I noted that our DNA is bDNA. There are actually other forms of DNA, other ways that two strands of nucleic acids using deoxyribose can bind together in a helix. These are denoted by A form, B form, and Z form. And you can see them pictorially here. A form generally is a more condensed form of the B form. It has the same type of rotation, except that there are more nucleotides compressed within one of these rotations. Z form is having a variety of differences between A and B form. You can see its rotation is actually the opposite. So it is right-handed instead of left-handed. And the binding that is occurring and the amount of nucleotides within a span is also very different. Here's a table to kind of break it down. Again, A and B are both right-handed, whereas Z is a left-handed rotation. And the amount of bases, the amount of these nucleotides in a turn differs between the three forms as well. Now, only slightly, you can see A has 11, slightly more, B has 10.5, and Z has 12, the most. You can also see how fast the helix rises per base pair. So again, this is essentially the height of that rise between 2 points per the number of base pairs there. And here is where you can see how A form is more condensed. There's a less rise per the number of bases when you compare it to B form. Similarly, the Z form has much bigger rise that gives these bigger gaps between the turns. Now, between the two strands of a normal B type DNA and DNA, we know that the chains themselves, the sequences within the chains, is what's called complementary. So again, C binds with G, etc. So two different chains binding together that are complementary toward each other. But one other feature is that they are also anti-parallel, meaning that one chain that is running from 5' to 3', red in that direction, binds with another chain that if you read from the top where it binds, it would be going from 3' to 5'. This is what we mean by anti-parallel. This is important because it does affect how DNA replicates. DNA replication, when we are looking at two different strands that need to then make a whole new set of strands, occurs first by just separating those two strands. So again, we have two nucleic acid strands that will start to separate. And each of those strands can then serve as a template for the synthesis of a new strand. This is because they are complementary. That is how they are able to serve as a template for the opposite partner. This type of synthesis, forming a new strand, is catalyzed by an enzyme that we'll be talking more about called DNA polymerase. DNA polymerase is actually a very large enzyme. So that means that it is a protein that is made up of multiple subunits. Remember, it has quaternary structure. In the end, after synthesis, because of this anti-parallel nature and the complementary nature, what we have when duplicating DNA is a new set of DNA, so two new DNA molecules that are made up of one daughter strand or one new strand and one parent strand or one old strand. You can see this visually in the picture based on the colors.